go and mix with the blood and reach the peripheral tissue. Liver does not require insulin for glucose uptake, so except liver, all other tissue, even brain also don't require. So wherever GLUT4 glucose transporter are there, those tissues require insulin for glucose uptake. Now those tissues, insulin will not enter the tissue. It will reach the tissue, it will reach the cell, it will trigger the cascade pathway, hormone receptor mediated cascade pathway. Right? After that thing is done, what thing is done? The work, the function of insulin is done, it should be degraded. Insulin is made up of what? Amino acids. Those amino acids will be poured back into the amino acid pool. What is the pool? In the cell where all the amino acids are available. For what? For synthesis of again a new polypeptide. Now this process is the healthiest metabolic process in our body. If anything goes wrong, say for example, there are certain compounds which trigger the breakdown of insulin. If some person develops such disease develops such conditions, not disease, the conditions where those substances are more active, what will happen? Their insulin will be produced but will be degraded at faster rate. So the body will sense there is insulin deficiency. Right? So these are called metabolic disorders. In a nutshell. Right? Now, if a compound cannot be degraded, that will get stored or accumulated within the cell. Within the cell, there is only one place where it can get accumulated. That is lysosome. That is why these diseases are also called under single canopy. একই ছাতার নিচে যদি ধরো সব ডিজিজকে তাকে বলা হয় লাইসোজোমাল স্টোরেজ ডিসঅর্ডার্স। লাইসোজোমাল স্টোরেজ ডিসঅর্ডার এর মধ্যে কি কি পড়বে? Diseases which are associated with glycogen metabolism, we have a separate name for them called glycogen storage disorders. Diseases which are associated with mucopolysaccharides metabolism, we have a separate name called mucopolysaccharidosis. Right? Diseases which are associated with different types of lipids, phospholipids, we call them phospholipidosis. Right? So this is the basics of any storage disorder. Why it is called storage disorder? Because the undigested product gets accumulated which has no function. Its biological function is over, which has no function any further. Similarly. So here, the first one, the GSD, type 1 GSD. What is the name? Von Willebrand's disease. Here, what are the clinical features we see? Hypoglycemia, right? Hepatomegaly, and lactic acidosis, and hyperuricemia, and hyperlipidemia. Right? These all are happening in a patient suffering from GSD1. Now, what all tests you will perform to diagnose GSD1? The first thing is you perform the resting hypoglycemia. The patient is not doing anything but still having bouts of hypoglycemia. When do you call hypoglycemia? When the blood glucose level is below 60. Below 60 is hypoglycemia. Right? Up to 70, it is normal, 70 to 100. Up to 60, it is tolerable. Okay, fine. That can be reverted back by giving some food. But below 60 means something is very, very wrong. Right? Case is serious. Right? So below 60 milligram per deciliter if we find any time of the day, that is hypoglycemia. So this is one. Number two, if there is hypoglycemia, then the cells will undergo anaerobic respiration. So anaerobic respiration will produce lactic acid. So lactic acid will accumulate in where? it has to be excreted through urine. Any acid is excreted through urine. So it will accumulate in the blood. So if the blood accumulates acid, what will happen to the pH of the blood? Acidosis. So this kind of acidosis is called metabolic acidosis. It's called metabolic acidosis. Fine. Now glucose cannot be utilized by the cell, but it can be stored in the form of glycogen. Glycogen breakdown process is hampered. What is the enzyme defect here? Glucose 6-phosphatase. So the last step of the glycogenolysis in the liver is not happening. That means glucose is unable to cross the hepatocyte and come into blood. Followed? But inside the hepatocyte, glucose is plenty. Correct? So what will happen? Recall the fate of glucose 6-phosphate. When there is no production of glucose from glucose 6-phosphate, then glucose 6-phosphate will go to production of fat, lipid. So there is hypercholesterolemia. There is hypercholesterolemia. Done? Now, come back to the next thing. What is that? Hyperuricemia. Now, this is very interesting. Hyperuricemia. There are many explanations given in many types of books. Here, I would suggest you to follow this explanation only. Rest all explanations are not so sturdy to stand in front of this. Correct? What is that? The lactic acidosis, that is accumulation of lactic acid in the blood, requires excretion of lactic acid through the urine. Correct? In the renal tubule, the lactic acid is preferred over another organic acid called uric acid. In the renal tubule, two acids compete for excretion through the same transporter, renal tubule. Correct? One is lactic acid, another is uric acid. So if the lactic acid is preferred for excretion, then who will be left out? Where? In the blood. So what will be the condition? Hyperuricemia. Now, what are the other causes for hyperuricemia other than type 1 GSD? That is overproduction of nucleic acid or over breakdown of nucleic acid, because the end product of purine catabolism is uric acid, which increase in case of gouty arthritis. Correct? Gout. So gout can cause hyperuricemia. Type 1 GSD can also cause hyperuricemia. Symptoms are same. Diseases are different. What is the difference? How do you differentiate? It's called differential diagnosis. Both are showing hyperuricemia. One will show hypoglycemia. Another will show normal glucose concentration. Okay? One point. Second point is the finding of lactic acid in the urine. If lactic acid is excreted more in the urine, then what will happen to the pH of the urine? Low. So find out the pH of the urine. Correct? pH of the blood is also a good marker. If there is hyperuricemia due to type 1 GSD, the blood will show acidosis, whereas that will be absent in case of gouty arthritis. Okay? Now come to number 5, McArdle's disease. Don't look at me. See that. McArdle's disease. What is the enzyme defect? Muscle phosphorylase. What is the symptom? The only symptom is the patient mostly are female. They will come to you and they will be very flabby, hefty. Okay? So first thing you will advise is do exercise and reduce weight. Again, on follow-up, she will come and complain something else. Doctor, I am trying to do exercise, but I get tired very soon. I start having muscle cramps. Correct? I start having muscle cramps. Mostly calf muscle. Correct? Now, how to diagnose? Remember, this is non-lethal. Patient will not die. Nothing will happen. She will be a little fat, that's all, throughout the life. Okay? She can't take exercise. She will walk slowly. You can't advise her to go for treadmill test because she will have muscle cramp. Problem is the lactic acid is accumulated within the muscle and it cannot escape. It cannot come out of muscle. Lactic acid is produced in the muscle, it will remain in the muscle. So if you test the pH, if we get tired out of rigorous exercise, then our blood pH will drop due to lactic acidosis. In these patients, type 5 GSD, the blood pH will not drop because lactic acid will be absent in the blood. It is accumulated in the muscle only. Correct? So this is how, see, 3 diseases you can differentiate. Correct? Type 2 and type 4, you need not to worry because they will not come to you. They will go to, okay? You need not to worry. That's all. So we'll go to now next one. It a close call. So now we are on the verge of carbohydrate metabolism. This is probably last but one class of carbohydrate metabolism. In the next class I will try to finish the carbohydrate metabolism. And you will finally, you will stop seeing my face. Huh? You'll be happy? No. Then doesn't matter who stands. Very good. So, the metabolism of fructose and galactose. First, we'll see fructose. The metabolism of fructose is very simple. What is that? The fructose metabolism takes place inside liver. Enzyme, fructokinase. Product, fructose 1-phosphate. This is a major pathway. Fructose, আচ্ছা আচ্ছা দেখে নে আগে পাসওয়ার্ড দেখে নে তারপর একটু পড়িস পরে আয়া কোথায় গেল পাসওয়ার্ডটা দাঁড়া Fructose has 2 pathways. One is major pathway and is minor pathway. What is the source of fructose? Sucrose, right? Any fruits? Yes, there are many fruits which are sweet in taste. They contain fructose. Say for example, mango. You like mango? You take mango? You take mango? Did you take mango ever? Thank you. I think, see, this is a syndrome of called fish eye syndrome. You know, the fish eye has no eyelids. They keep the eyes open. People always think the fish is looking at me. Actually, fish is sleeping. No, sir. No, sir. Okay. So when you do not respond to the question properly, I feel you are having fish eye syndrome. Yeah. So the fructose goes to liver first. Fructose metabolism is very, very important. In the liver, it has 2 options. 1, the major pathway. 2, the minor pathway. What is the minor pathway? Hexokinase. Does liver has hexokinase? Does liver has hexokinase? No. Some other tissue might have hexokinase, like intestinal tissues. Correct? So in the intestine, liver has no hexokinase. So in the liver only fructokinase. Fine? So that's why you might have, if you see the diagram in many books, in the same page they have drawn the major and minor pathway. So the major pathway takes place in liver by fructokinase. The minor pathway of utilization of fructose occurs in extrahepatic tissues via hexokinase. Hexokinase produces fructose 6-phosphate. After that, it follows the glycolytic pathway. But a major pathway produces fructose 1-phosphate, which is new. To break them, we need one aldolase. It's called aldolase B. Aldolase B. A and B. Aldolase B cleaves fructose 1-phosphate into gap? No phosphate, only glyceraldehyde. গ্লাইকোজ কি ছিল? গ্যাপ ছিল, পি ছিল, পি নেই এখানে, খালি গ্লিসারaldehyde আর dihydroxyacetone phosphate. Fine. From dihydroxyacetone phosphate by team we can produce this one and we can continue glycolytic pathway. Right? But glyceraldehyde need to get phosphorylated. So triose kinase will phosphorylate glyceraldehyde to glyceraldehyde 3-phosphate. Again this product is formed. Same product, then it will enter glycolysis. So we need to remember 3 enzymes. Fructokinase, it requires ATP, magnesium ion, then aldolase B, it splits the molecule into 2 half, glyceraldehyde and dihydroxyacetone phosphate. The DHAP goes to glyceraldehyde 3-phosphate by triose phosphate isomerase and enter into the glycolytic pathway. The glyceraldehyde is phosphorylated to glyceraldehyde 3-phosphate via triose kinase in presence of ATP and magnesium ion. Glyceraldehyde 3-phosphate enters into glycolytic pathway. But problem arises the entry point of fructose metabolism. Where they are actually entering? They are entering beyond the regulatory point of glycolysis. What is the pacemaker of glycolysis? PFK1. So PFK1 step is being escaped by fructose. See, it is entering glycolysis beyond the main regulatory step of glycolysis. That is PFK1. Phosphofructokinase 1. So what will happen? It will no longer be regulated by the hormones which regulate glycolytic pathway. So what will happen? It will go on producing pyruvate. Pyruvate will produce more and more acetyl coenzyme, and acetyl coenzyme is not required, is excess. Then what will happen? It will produce fat, cholesterol. Correct? Dihydroxyacetone phosphate is a initial or precursor for phospholipids. Glycerol 3-phosphate is a precursor for phospholipids, triglycerides. Glycerol 3-phosphate will be produced from glyceraldehyde 3-phosphate via dehydrogenase. So these products will trigger the formation of fat of different types: phospholipids, triglyceride, cholesterol. So what will happen? Hyperlipidemia. That is why when you go fat, the doctor says the first thing is cut your sugar out. Don't take sucrose. Sucrose is not harmful. Fructose is harmful. Don't take fructose. But they never say don't take mangoes. Why? Suppose this is unusual that you are having only mangoes and nothing else in your diet. How many of you do that? Morning 5 mangoes, afternoon 10 mangoes, then evening 5 mangoes, in night again 20 mangoes. Like this diet, anybody is following? Ever followed? No. So what we do? 1 or 2 mangoes a day. That's all. Or in some days 4 or 5 mangoes for whole day. Next day again chicken biryani. Back to biryani. Correct? Fruit we like only for 1 or a couple of days. That's all. After that we back to biryani. We feel like kitna din ho gaya biryani khaye. So how much fruit we take? Not much. But why they do not say no to fruits which contains raw fructose? Because the fruits itself, not juice, fruit will contain a lot of fibers, which reduces the entry of fructose, which contains a lot of vitamins also. But when you take fruit juice, the fructose gets concentrated. Correct? The uptake of fructose is more high. Correct? That is why they say that carve out sugar from your diet in order to reduce weight, triglyceride particularly. Right? Now we'll see the different aspects of this metabolism. So it is in the liver by fructokinase first, and then aldolase, which I have shown. They bypass the steps of glucokinase and PFK1 because they enter through the DHAP step to the glycolysis. So there is no regulation of glycolytic pathway over the fructose metabolism. Okay. Now, the affinity of aldolase B for fructose 1,phosphate is much poorer than the fructose 1,6 bisphosphate. Thus, fructose 1-phosphate accumulates in the fructokinase expressing tissues, that is liver. Muscle contains only hexokinase, can phosphorylate. Muscle means extrahepatic tissue, EHP, extrahepatic tissue. So they can metabolize this one via hexokinase, whereas liver contains no hexokinase, only glucokinase. Fructose 6-phosphate goes to formation of pyruvate via glycolysis. If fructose 6-phosphate accumulates, then they produce these products. Where it occurs in extrahepatic tissue. Fructose 6-phosphate. There are many products which require fructosamine, extrahepatic tissue. One such tissue is testis. You do not have, but 50% of the population have. Right? And we are here because the fructose were present in the semen. What is the function of fructose in the semen? It is still not known clearly, but it is found that a healthy individual must contain a good amount of fructose in the semen. That is being tested also regularly in the laboratory. We receive different semen samples for the testing of the same fructose level. If the fructose level is low, patient is advised to eat fruits to increase fructose level. How far it helps, God knows, but it is advised. So these fructose goes for production of fructosamines, then UDP-glucose, N-acetyl glucosamine, all these products. So they produce proteoglycans, they produce glycolipids, they produce glycoproteins. All this happens outside liver. First it forms, glucose and then sorbitol. Aldolase reactase forms fructose to sorbitol forms glucose. The conversion between glucose and fructose has an intermediate called sorbitol. Okay. Glucose to sorbitol to fructose. Fructose to sorbitol to fructose. This particular reaction might take place in testis. which is the reason for what semen contains, the seminal fluid contain high content of fructose. Deficiency of fructose correlates with male infertility. এটা the sperm needs glucose. According to this line, fructose is responsible for production of glucose in the sperm. So when the sperm outside the body, how will it survive? So the sperm will die prematurely. So it will never reach the ovary. Before that, it will die due to starvation. So what will happen? There will be no baby. Right? And patient will come to you. If they do not have baby, they will come to you. If they have baby, they will come to you. During having baby, they will come to you. After baby, if baby wants to become Baba, then also you. So they will come to you. Don't worry. You all have passed that stages. Now diseases. Two such diseases are important here. One is essential fructosuria, which is due to deficiency of fructokinase. Simple. Fructose gets accumulated in the blood. Excess of fructose cannot be metabolized, so it gets excreted to the urine. What is the test you will perform to find out fructose in urine? Write down. Selivanov test. This is a test done for identification of fructose in a given sample. So what will happen? When we get semen sample for analysis, we centrifuge it, and then we take the liquid part, and then the liquid part, the seminal fluid, liquid part. The liquid part we test by this test to find out whether it has good amount of fructose or not. If the test fails or response less than normal, then this fructose is insufficient. Right? So that could be a reason why the sperm is dying prematurely. Now that is the reason can be reason for infertility. See, this is fructokinase deficiency doesn't pose any serious condition. Urine will contain fructose. That's all. You will go a little more frequent for urination. That's all. But you will not die. Nothing will happen. The second one is dangerous. Second one is dangerous. That is deficiency of. Oh no, not this one. Sorry. Eight. This one. This one is dangerous. This one is dangerous. It's caused due to deficiency of aldolase B. So what will happen? Fructose 1-phosphate will accumulate. It will go on accumulating. The problem is it will damage. Fructose 1-phosphate is osmotically active. So it will draw more water inside the liver, hepatocytes, and hepatocyte will swell up. After some time, it will stop swelling because it will die. Right? So what will happen? If the same thing happens in the kidney, right? So these two organs are affected, and you will die due to liver damage. So aldolase B deficiency will cause liver damage, cirrhosis of liver. cirrhosis of liver. It is hereditary fructose intolerance, HFI. Hereditary fructose intolerance, HFI. It has many subtypes. I am not discussing those. Okay. Now the third one, the less important is this. Less important. Okay. At least you remember these two. First one, essential fructosuria, and second one is HFI, hereditary fructose intolerance. Now we come to galactose metabolism. The galactose, from where we get galactose? Bolo? Lactose. Lactose is a milk sugar. So milk sugar contains lactose. Lactose contains galactose. We get galactose. Very good. But problem is galactose metabolism poses a lot of problem. The site for galactose metabolism is liver, as usual. Liver has taken all the pain to metabolize everything. If we drink alcohol, it is liver. If we do not drink alcohol, eat biryani, it is liver. If we do not take biryani, if we do not take alcohol, if we take only mangoes, it is liver. If we do not take mangoes even, if we take only paracetamol, it is liver. Right? So the liver takes a lot of pain to metabolize and to get rid of things from our body. Very good. So galactose gets metabolized in the liver by an enzyme called galactokinase. This is very, very important pathway. Very often, almost every year there will be question from here. Almost every year, if not every year. প্রত্যেক বছর এখান থেকে কোশ্চেন থাকে, থাকবেই, পড়তেই হবে, মাস্ট, একদম। গ্যালাক্টোজ ফার্স্ট ওয়ান হচ্ছে গ্যালাক্টোকাইনেজ, এটা হচ্ছে মেজর পাথওয়ে। মাইনর পাথওয়ে বাই হেক্সোকাইনেজ, not found in liver. We do not worry about that. We worry about the hepatic pathway. Hepatic pathway is operated by galactokinase. It requires ATP, it requires magnesium ion, it forms galactose 1-phosphate similar to fructose. Fructose has fructokinase, fructose 1-phosphate, galactose has galactokinase, galactose 1-phosphate. Same. Next is different. This galactose 1-phosphate is converted to glucose 1-phosphate. How? Not directly, in an indirect way. How? See this side pathway. The same hepatocyte contains glucose 1-phosphate. From where it gets glucose 1-phosphate? Glucose to glucose 6-phosphate by glucokinase. Glucose 6-phosphate to glucose 1-phosphate by phosphoglucomutase, the initial step for glycogen synthesis. Right? So glucose 1-phosphate is then converted to UDP-glucose. It's the same pathway for glycogen synthesis. Up to UDP-glucose, it will be same. Then this UDP-glucose, if it is not synthesizing glycogen, then it will go and exchange this galactose. UDP-glucose will take up galactose from galactose 1-phosphate. It will become UDP-galactose, and this glucose will be left as glucose 1-phosphate. Then the reverse reaction, glucose 6-phosphate, and it goes to glycolysis. But the galactose which is taken up by the UDP-glucose in exchange of glucose now will be converted to UDP-glucose via enzyme 4-epimerase, because galactose and glucose are epimers at C4. If we just reverse the OH position of C4, then glucose will become galactose or galactose will become glucose. So 4-epimerase is the enzyme which converts UDP-galactose to UDP-glucose. The enzyme cannot act on galactose alone. It must be associated with UDP because UDP blocks the C1, the more reactive carbon atom. Otherwise, it will exchange C1. Correct? So 4-epimerase can function because of UDP. Without UDP, it will not function. So the steps are very easy. Galactose, galactokinase, galactose 1-phosphate. In exchange with UDP-glucose, it forms UDP-galactose, UDP-galactose to UDP-glucose by 4-epimerase. Enzyme here is UDP-glucose, galactose 1-phosphate, uridylyltransferase. Name of the substrate, first, UDP-glucose, galactose 1-phosphate, uridylyltransferase. UDP-glucose, galactose 1-phosphate, uridylyltransferase. Correct? Any question? No question, good. Now, galactose metabolism. So there are two enzymes here. What are two enzymes? One is this uridylyltransferase, another is galactokinase. If these two enzymes are deficient, then there will be a problem. Galactose will accumulate in the liver. If galactose accumulates in the liver, what will happen? Hepatomegaly, liver damage. Same process because of fructose. So UDP galactose, sorry, I can ask you. So galactosemias, দেখো কি লিখেছে galactosemias লেখা আছে কেন galactosemia হচ্ছে এটা common name for diseases due to absence or deficiency of a number of enzymes. Correct? If it is galactokinase, then severity will be somewhat different. If it is uridylyltransferase deficiency, then severity will be somewhat different. If it is 4-epimerase deficiency, then severity will be least. Higher the enzyme affected in the metabolic pathway, more severity will be seen. Right? But here we'll see this particular enzyme, these two enzymes are more important for galactosemias. The basic thing is galactosemia. If you ask, if this question comes, what is galactosemia? You must say galactosemia is a condition, generalized condition, which shows defective galactose metabolism in the body. Correct? It is characterized by high level of galactose in the blood, sometimes in urine. High level of galactose in the blood and sometimes in urine, called galactosuria. How do you test galactose in urine? Remember, in urine, we perform twice. We firstly perform Benedict test. It will come positive because galactose as well as glucose both are releasing sugar. Correct? Up to here, it will be shown for galactose, Benedict test will be positive for all the three: glucose, galactose, fructose. Now we need to find out which sugar is present. So we have test for fructose. If we perform Selivanov test and Selivanov test is negative, that means it is not fructose. Maybe glucose or galactose. Then we perform glucose oxidase test. If it fails to answer, that means it is not glucose. Is what? It is galactose only. It is galactose only. Both will show hypoglycemia. Fructosuria and galactosuria. Okay? Now it must, both will show hepatomegaly, fructosuria as well as galactosemia. You cannot differentiate from that. Right? But you can differentiate by checking the eye. If you have unilateral or bilateral, mostly bilateral cataract. Sometimes the disease is so severe, the baby is born with cataract. The baby is born with cataract, bilateral cataract, both the eyes. Newborn baby with cataract. In case of severe form of galactosuria or galactosemia. Okay? Cataract is due to what? Formation of galactitol. Glucose and fructose give rise to this polyol. This is called polyol. It's multiple alcohol. This has got a name called sorbitol or sorbitol. The corresponding polyol formed from galactose is called just nomenclature. You must know the names. And these polyols usually form in the eye lens cells, and they get drained from the lens. But if a person suffer from galactosemia or diabetes mellitus, type 2 diabetes mellitus, then what happens? The glucose concentration will be high or galactose concentration will be high. So the rate of production of polyol will be high. If it surpasses the rate of excretion or drainage from the lens, what will happen? It will start, gets accumulated in the lens. Slowly, it will swell up lens and will damage the normal architecture of the lens. Then what will happen? Bacteria will infiltrate. They will feed on these polyols. They will produce opaque materials, leading to cataract formation. If you do not take care of this, you can become permanent blind. Okay? This is the mechanism for cataract formation by polyol pathway. So they follow polyol pathway for cataract formation. This is a sorbital pathway. Sometimes they ask sorbital pathway because it is the only reason through this pathway is actually diabetic cataract or cataract can be seen. Glucose to sorbitol fructose. So glucose to sorbitol or fructose to sorbitol. Whatever will be more, if glucose is high, then diabetes mellitus, it will form sorbitol. If fructose is high, it will form sorbitol. If galactose is high, it will form sorbitol. But there is only few sorbitol can escape from the lens. Body can cope up with small amount of sorbitol production. If you produce in huge amount, then it will gets accumulated in the lens. Now, this particular pathway is called remember I almost covered many tests, many differential diagnoses. The same question will be asked in the examination, I tell you. Be very cautious. Same question. Same thing we will teach you throughout the year. And same question will be asked in the final. Now, the last pathway. The last pathway for carbohydrate metabolism. This is glucuronic acid pathway or uronic acid pathway. Uronic acid pathway. It starts with glucose to glucose 6 phosphate. Right? Glucose 6 phosphate to glucose 1 phosphate via PGM, phosphoglucose, glucose 1 phosphate to UDP-glucose via UDP-glucose 1 phosphate, uridylyltransferase. Right? Then UDP-glucose to this enzyme is drink water. For 2 minutes, drink water. Last 2 minutes, drink water. UDP-glucose undergo oxidation, undergoes Boswell. UDP-glucose undergo oxidation to form glucuronic acid. Aldehyde oxidized to form uronic acid. The C1 is oxidized. If C6 is oxidized, we call gluconic. Sorry, C1 is oxidized, we call gluconic. C6 is oxidized, we call glucuronic. If C1 and C6 both are oxidized, we call glucaric. So first one is attached with UDP. The first carbon cannot get oxidized. Only 6 carbon will get oxidized. Otherwise, which has more tendency to get oxidized, C1 or C6? C1, because it is halfway to oxidized form, aldehyde, carbonyl form. Alcohol, carbonyl group, then aldehyde, then acid. So it is only halfway there to oxidation form. So that's why it is blocked by UDP. So that C1 is not get oxidized. Only C6 will oxidize. And that is done, that is possible only via enzymatic process. Glucose on the action of bromine water, mild oxidizing agent, forms gluconic acid. Glucose in presence of strong nitric acid, which is strong oxidizing agent, forms glucaric acid. But no such chemical reagent can convert glucose to glucuronic acid. Why? Because C1 is open. So C1 is blocked by UDP prior to oxidation, so that we can produce glucuronic acid, which can be used for synthesis of what? Mucopolysaccharide, etc., 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 glycolipid, glycoprotein, so many things. Correct? All this pathway here see some enzyme is there. Block in pentosuria. Block in pentosuria. Name of the enzyme, write down. Xylitol dehydrogenase. Xylitol dehydrogenase, pentosuria, essential pentosuria. Enzyme defect, xylitol dehydrogenase, quickly. Xylitol dehydrogenase. Okay. Lab finding, pentose sugar in urine. Lab test, लिखे ले हाथ चला. Lab finding pentose sugar in urine. Lab test. Bial's green test. Bial's green. BIAL. Bial's green test. है ना? Test for ribose sugar, মানে pentose sugar mostly. হলো? এগুলো essential pentosuria. তারপর লেখো humans and primates. Humans and primates. Humans and primates. And guinea pig. Cannot synthesize vitamin C. Cannot synthesize vitamin C due to absence of enzyme. L-ketogluconate dehydrogenase. L-ketogluconate dehydrogenase. L-ketogluconolactone, sorry, lactone. L-keto, capital L, লিখে দেবো? L-ketogluconolactone dehydrogenase. কোনো বই অক্সিডেজ লেখে L-ketogluconolactone oxidase, একই ব্যাপার, অক্সিডেজ লিখতে পারো, ডিহাইড্রোজেনেজ লিখতে পারো। L-ketogluconolactone oxidase. অক্সিডেজ কোনো বই ডিহাইড্রোজেনেজ দেওয়া থাকে, কোনো বই অক্সিডেজ দেওয়া থাকে, যেটা পাবে লিখো, অসুবিধা নেই, both will give you marks. Okay, তাহলে এইটা থাকে না বলে we cannot synthesize ascorbic acid, so we have to rely on citrus fruits for ascorbic acid, vitamin C. Otherwise vitamin C खाने का जरूरत नहीं पड़ता, है ना? That's all. Thank you and good night.